Thank you for a kind introduction. I will show again this magic room, Viktor, and I can only add as a comment that due to my dimensions, I was fit into this room alone with Viktor. If some people could manage to have two. So our Honorable President Soomere mentioned before soft power of science. And I think in my memories about Viktor, this is just the soft power of science which characterized Viktor for me. And I mainly learned from Viktor that if you love something and if you do something about it, then it's always interesting, at least in science. And that's why I'm very thankful that I met Viktor in my life. And this happened around 1987 when I managed to get to Sweden to Professor Tomas Hökfelt's group. And of course, Tomas introduced me Viktor immediately. And the main contact with Viktor was in this room. It was a time when you had to go to a library, good library, to copy the scientific articles. And we always went to Karolinska Institute and copied something. And then after that, I took a liberty to go to Viktor's room and discuss things with him. And he was always interested. And his very characteristic feature was that he was talking about something, and when he found somewhere down in a pile of articles, this article which was just the right one in this case. And that was very impressive for me. And the Galanin studies were mentioned here several times, and I was starting with Galanin. I was lucky. I think Galanin was just discovered 4 years ago, and I was on this. And you can see here there are 3 receptors, and there are several biological activities of this neuropeptides, which makes it very, very interesting. And I titled my talk today, Beyond Viktor's Research. And actually, previous lectures here today were always beyond Viktor Mutt's research. So we are talking about the same thing. What has happened after Viktor discovered these 50 peptides or more. And that is what makes science excellent, that it's ongoing, it doesn't stop. You find new avenues, you find new ideas, you have fantastic things which are very far from original discoveries sometimes. And we started from Galanin research and regular Galanin synthesis. I also tried some collaboration with Viktor by making or trying to send him from Tartu to Stockholm 2 tons of porcine intestine in frozen state in 1988. I don't know if you younger people, if you understand exactly what I'm saying, but 2 tons of intestine by ship in Soviet time from Soviet Union to Sweden. Oof. And that was not a very successful project. Viktor paid for these 2 tons of material, but it was only 15% of activity of these peptides which he separated from them, and we stopped that. The problem was that in slaughterhouse you have to separate the intestines and wash them intensely. And we paid for these slaughterhouse people, of course, for that. But they took the payment, but they didn't wash them very carefully. So we sent 2 tons of shit in summary. That happens. And later, Rannar Sillard, who was working in Viktor's lab, succeeded better to separate sheep brain in Võru slaughterhouse, right? And this worked, and we were able to together to publish the sheep sequence as well. But in our lab, we were thinking about developing ligands for galanin receptors, which are 3 receptors. When we started, it was only 1 receptor known, then 3 receptors became available. But we started from rat brain receptors, and then we knew from NMR what the structure of galanin was on the left. We know by doing studies together with Tiit in Tamas's lab, we were able to define the pharmacophores. And then we got the idea to make chimeric ligands, where we have at N-terminus the N-terminus of galanin, and at C-terminus different sequences from other neuropeptides which were known to have activity at C-terminus. And they were, some of them are listed here. You can see some are combinations of 2 neuropeptides, and some are not. Some are just galanin part connected to fantasy sequence, alanine leucines. You can see M871 here also, which I will show you a bit later. They were fantastic galanin receptor ligands. They contained shorter fragments of neuropeptides, but these neuropeptides or these ligands were recognized by both components, both neuropeptide receptors. And that was a synergistic effect, and that seemed fantastic because the improvement was like 1,000 times. So we believed that they bound to both receptors simultaneously. And I took this example, which is, again, Tiit is involved here. This is from '92, more than 30 years ago. And we presented in this journal this very naive idea that these chimeric ligands could simultaneously bind to both receptors. And it is basically, this paper is unnoticed. It has around 100 citations only in 30 years. But what happened after that, and in parallel with this work, with this insignificant work, was that dimerized receptors became evidently available. Multimerization has been indicated today. And if I have a good day, then I can say that we also made some contribution to this. But now with several receptor mosaics and things like that are available in many examples. And here is one example of later published to 2023 when such a thing was published most recently. Now, if we look at these ligands, then we made, I think we made hundreds of these ligands. Later, Runner was also involved in synthesizing them because the colony receptor subtypes became available, and we tried to contribute into this field, having these receptors expressed in cell lines. And some of them are shown here in this figure. On the right side, you can see two non-peptide ligands. It was a question about non-peptides, mimetics. And Kalnon and Kalmik is from Tomas group at Scripps, I remember. And Kalnon we made together in Stockholm. Small molecules which have maybe micromolar activity only, but which showed very, very interesting effects. And we tried to publish the Kalnon, at least in Nature Medicine. And after fifth round, we got rejection. One referee had a very good argument. He said that it will never be a drug. I don't know where he knew it, but that was a response, and that it was rejected finally. But there are several nice publications with these peptides, and they have their future ahead, I think. And now, ending this topics, I found a very recent paper with our M871, which I showed you before, which is based on galanin, and it's an antagonist. We made many experiments with that. Other people basically did this experiment, but I found this where it is a galanin receptor 2 selective ligand, and it has a role in craniofacial development. Could anybody like Viktor or somebody in early days predict that we find this kind of effect? I have no idea. Yes. Yes. And it's just an example in an event where it will find itself, this study or research. But then I very briefly will talk about drug delivery, because we have been using many novel peptides for drug delivery, for all types of drug delivery, which is very interesting. And this topic is interesting because the size of efficient drugs becomes larger and larger nowadays. You can see here a comparison of 2007 and 22, and without looking at the names on this, you can see that the molecules of drugs have become much larger. And this is a challenge for drug development. You need to use special delivery vehicles to get these drugs over the barriers to the cells, or over the blood-brain barrier, or whatever barrier. And you can see the sizes approximately where the cell therapy is not characterized by molecular mass, but these sizes are very well known, and this can become very big. Now, many delivery vectors are available there. Some of them are listed here, and they can be peptides, or they can be non-peptides. They can be different kind of molecules, and many of them are. And the cargos can be also all kind of molecules: peptides, proteins, nucleic acids, small molecules, and even cells. And the issue is to get them through these barriers into the cells sometimes, or into the targeted organs in a body, because we're talking about the drugs, and we are interested in taking the drugs into this target. For example, cancer. We don't want to kill other tissues than cancer cells. And this is a tricky thing, because we mostly don't understand what we want to do and how to do it. In vivo is extremely difficult today. We get inflammatory and immunogenic responses, and we have to improve the targeting. And that leaves a huge area of research to solve these problems. And that's why we are concentrating on one piece of this, and this is cell-penetrating peptides, or peptides, which are short peptides. And I also listed here that several groups in Estonia are also involved in CPP research. And these peptides are many. I listed only a few of them. In red frames are ours, but there are really thousands of them available today. And our peptides are based again on these chimeric galanin-based peptides, which I showed you before. And basically, the best lead was transportin, and transportin 10, which you can see, and in a sequence, you can see the galanin-based sequence. So they contain a part of galanin, and in the C-terminus there was a mastoperin, a wasp venom toxin peptide. And when we put galanin together with this mastoperin, we got an incredibly interesting peptide for some reason. It was by chance. We got this peptide, which entered the cells when incubated in small quantities together with cells. And we have also now hundreds of these peptides available. PepFacts and NICFacts are listed here. You can see there is always a short galanin sequence available. And this is something which I don't know if this is relevant at all, but we still use this sequence. And what we can do with these peptides, we can use the connection of different cargos by covalent or non-covalent strategy, and we can use them for delivery of nucleic acid, basically, but also the other types of molecules. And we can, by adding homing sequences, for example, target them to different tissues. And here are these examples for gene silencing and gene therapy, which they have a great potential for. And I show you a few pictures about it, where our PepFact is connected together with the oligonucleotide, and it forms nanocomplexes, and in electron microscopy you can demonstrate this uptake of with nanoparticles. And in vivo injected, here the siRNA is injected to knock down the luciferase protein. You can see that knockdown of a fluorescent protein was achieved. And here is one example of using tumor targeting, where we put the target peptide together with our CPP, and we achieve tumor delivery of an animal model, and also we are able to show tumor growth inhibition in some cases. And final illustration is about brain delivery. It seems that in case of in some vectors, we are able to label the brain tumor after IV injection. It's a glioma targeting, so we called it G-HOPE peptide. And I think this will go further and further. This research is not a dead end, it's just opening, I think. I think it will continue until we have all good drugs based on such delivery methods. Of course, I can't guarantee that something very new and exceptional will come up, but then it will be then. That's it. Thank you.
So, thanks. Now, time for questions. We have time. Yeah, Martin.
Thank you. Thank you, Ülo, for a very interesting lecture. What do we know today how CPPs enter the cells? Do we know the mechanism of internalization?
Yeah, there are several mechanisms, and one of them is, which is often available for short peptides themselves, and this is that they will go through a plasma membrane by membrane interactions, charge-charge interactions. But in most cases where you try to deliver the cargo, like bigger molecules, then endocytosis takes over. And in that case, endosomal escape is the most important thing. And what we believe is that our peptides are, we have been lucky that they have a good endosomal escape. If we talk about plasmids and we want to take them into a nucleus, we need only a few copies in the nucleus to start it. So it's not maybe so. But if we talk about our recent discovery of mRNA transporters, which should be in cytosol, then we have even peptides available which are very efficient in such delivery. So it's very different for different peptides, this mechanism. But I think these two basic mechanisms are in parallel.
Okay. Yeah, let's go.
If you figure out the two-dimensional structure of galanin, you put it like victory-like. Like V.
Sorry, what?
We put it like V-like or victory-like.
What is the structure of galanin?
Galanin structure, you put it like V-like.
It's like a horseshoe structure I showed you in one figures, which is based on NMR studies. But of course, every peptide is very flexible, and it depends on environment. And later, Astrid Gresson showed that But if we put it in STS mice cells, it's again the same structure as it was shown by NMR structure in water. But I guess it's very, very flexible, and depending on interaction partners, it will change.
More questions?
Okay, thank you.
No, thank you very much.